Welcome to the Chapter 14 wrap-up, where we walk on the wild side of non-Mendelian genetics. Prior to this, we've discussed complete dominance, which is what Mendel had discovered, where ultimately if you have even a single dominant allele, you're the dominant phenotype. But that's not always the case. As we looked into more complex traits, beyond just the P traits that he happened to look at, we see a bunch of situations where this is just not true. So the first two are often confused, so pay close attention. Incomplete dominance is going to be a lot like if you're mixing paint, where they're going to blend. And so that's this scenario here, where if you're homozygous for red, you're a red flower. If you're heterozygous, so one red allele, one white allele, you're going to be pink. And sometimes they'll use W instead of R. You don't technically have to use a lowercase r here because it's not completely dominant, so don't be surprised if you see like a W there. But those will be pink, so halfway between red and white. And then if you're homozygous for the white allele, you will be a white flower. So this allows us to get these mixes, where a tall and a short should give you medium, where a black and a white should give you gray. So incomplete dominance is like paints. It's like when you're blending. All right. Blending apparently is true occasionally at least, so the blending hypothesis people can rejoice. Now, codominance is going to be different in the terms that you essentially get both. And so this would be where if you have a red flower and a white flower, you would get red and white. This one can be seen when you've got something like sickle cell, where if you're a heterozygote, you'll have some sickle-shaped cells. That's these guys. They're the oddballs. And then you have normal-shaped cells. So if you're a heterozygote, you have a mix of both, which in this case is the best scenario, because it helps make you resistant to malaria, which is a good thing, especially if you live in parts of Latin America, Africa, anywhere where you would see that malaria is common. But it's not where so many of your cells are sickle-celled, where it leads to the other complications such as anemia, because the sickle cells don't live as long, such as blood clotting, organ failure, a whole bunch of nasty stuff. And so sickle cell is a codominant trait where you can essentially have all normal red blood cells. You can have a mix, that's the heterozygote, or you can have kind of all sickle-shaped cells, which is the worst situation. So if you're homozygous for the sickle condition, you get sickle cell anemia or sickle cell disease. That's the people that you see that have the issues with this, is because they are homozygous for that trait. The heterozygous individuals have a mix, they're actually the healthiest. That's why this has been selected in places in like Africa and Latin America where malaria exists. And then you've got multiple alleles, which are just scenarios where we don't have just an either-or proposition. So it's not just red and white, or it's not just normal red blood cells and sickle cells. We now have multiple options. And so blood types is the common one we use because we have several different blood type possibilities: A, B, AB, and O. And these will come down to three different alleles. We have an A allele, a B allele, and an O allele. And they'll typically be written I, A, I, B, and lowercase I. And the reason for this is A and B are both codominant. And so we use this I just to show it's an immunoglobulin. That's just the name, the fancy term for these protein pieces that are sticking off, the antigens. And so we're going to call it an immunoglobulin. And so the immunoglobulin version A, there you go, B, there you go. And then this O guy is written as a lowercase with nothing there because ultimately this one lacks the antigens. It's not making stuff. And it's also recessive to both A and B. So by making it lowercase, we can see that it's recessive. So the way this works out is you could have someone that, for instance, is AO as their genotype. Somebody else could be BO, unfortunately for them, I guess. And if we go through and do our Punnett square, you'll see that we can actually get every possible blood type from this. We could get an AB individual. We could get a B individual who's also a carrier or a heterozygote that has the O allele, so they could still have offspring that are O. You can get an individual that is A and a carrier, or you can get an individual that's blood type O. And so this was one that was used a long time ago. Just by looking at blood type, they could use it for basic crime stuff where it didn't narrow it down completely, but it could rule out certain suspects if they didn't have the right type of blood. They could do this as a very crude paternity type test where, once again, there's like multiple percentages of the population would have your type of blood type. But in this case, it at least does eliminate some individuals, so it could be used to kind of whittle things down a little bit. And the reason this is such a big deal for us is that if you have this blood type A, you have this molecule on the surface of your red blood cells. And so if I give your blood to someone who has blood type O, they don't have it, so they'll actually have antibodies that attack that blood because they will go after that A antigen. If I have someone who has type B blood, the same thing would happen if I gave that person's blood to a person who had type A or a person who had type O. Because they don't normally have that B antigen, they will have antibodies they produce and they'll attack it. So this is why O is considered largely the universal giver, because they lack all these antigens, so they can give blood to pretty much everybody else. Whereas the AB individuals, they have all the antigens, so they're the universal taker. They can pretty much get blood from anybody else. Now there is much more complexity than that. There's other blood type, I don't know what you want to say exactly, but there's other markers with blood type. One of the most common ones is the Rh factor, which is normally written as a plus or a minus. Plus is dominant, the minus is recessive, it's completely dominant, so that's why most people are positive. But there is another situation like this where ultimately if I give positive blood to a negative person, they'll also reject it. And it's kind of interesting because if you're a mother who happens to be negative blood and the baby is positive, you can actually send antibodies across the placenta and kind of like attack your child to make sure you start the abuse early. They can give you drugs to prevent that, and it only happens in that scenario. None of the other blood types cause this. But just be aware that we are just going over the basics of blood type. There are additional markers beyond these. It's just the common ones are ABO, and then the slightly less common but still frequently used thing that we talk about is the plus and minus, the Rh factor. So that's one case of multiple alleles. It also has codominance and complete dominance all mixed in. Now, next we've got some more odd situations. So pleiotrophy, this is just where one allele, so in this case we'll say sickle cell, doesn't just give you one phenotypic effect. It doesn't just say this is what you are. It doesn't just say here, have some sickle cell shaped, blood cells. It also says have resistance to malaria. It also says if you have, well, you're a homozygote for it, it also says have some extra clotting that you really don't want. Have your cells die earlier, so ultimately you're low on red blood cells, so you have anemia, which is just a lack of red blood cells, which leads to being tired, difficulty getting oxygen. It's going to say go ahead and have where these cloggings can cause pain and organ failure, and ultimately up to and including death. And so you get a whole bunch of different phenotypic effects, even though we're just talking about one allele for one gene. So this is essentially one gene, many effects. Epistasis is where one gene can essentially shut off another. So I like to think of this as like a circuit breaker in a house. You can have where the lights are all on in your room, and I can not turn those lights off. I can leave the switches all on, go down to the basement or wherever your circuit box is, and flip the switch to kill power to the whole room. So even though the switches are on, the lights are supposed to be on, they don't work. The lights go out. And that's how epistasis works. So for instance, when you look at like Labrador retrievers, they have chocolate and they've got black colors. That's the alleles they possess. So all labs are coded to be either chocolate or brown when you look at their alleles. Those are their options. However, there's another gene at a separate locus, a separate spot on the chromosome or in our DNA that can control whether or not we use those alleles. And so if you have that gene that happens to be on, we'll say, in most cases they tend to be recessive, so if you have two recessives for that other location, that separate location, it can essentially turn off the pigments. And so that's when you get a yellow lab. A yellow lab is still coded to be a brown or a black lab. It's that other gene just makes it shut that down. This also happens with albinism, where albinism just tells the other pigment genes no, and it just blocks them. And so because you don't make the pigments, you end up being colorless or having altered coloration. But it's not like there's any specific code that says make a yellow pigment. It's just a lack of expression of the brown or a lack of expression of the black pigment. So that's epistasis, that circuit breaker idea. And then polygenic inheritance is just where a lot of phenotypes are not controlled by just one gene. I like to think about this where you go to the store, if you've ever seen them actually tint paint, like for your house, they put it in the machine and it just adds, like to white paint, it adds little bits of different colors and different amounts called tints. And then after that, they shake it all up and you get your final color, which is whatever shade you wanted of yellow or blue or brown or whatever. But that's kind of how polygenic inheritance tends to work, where you have a bunch of different genes that each say, all right, let's say it's skin tone. That's covered by multiple genes, probably 10 or more. And so for each of these 10 genes, you have alleles that might say, let's not add too much pigment, or let's add a lot. And then you add all that together and you get your overall skin tone, which can go from translucent practically to very dark, depending on which ones you have. This is also why the children don't have to be the same as the parents as far as skin tone, because it all depends upon which version of their, like which allele they give for 10 different genes. So you could have where parents could happen randomly to give the lighter, if you will, the ones that say less tint, for each of these genes to one offspring, and they could give the darkest versions they have essentially to the other. So there are some kind of rare cases where you have parents that have children that are like really, really, really light and really, really, really dark just by probability, just by the odds of it. It's rare, but it can happen. And that's because it's polygenic. And you can normally tell something's polygenic because you see a spectrum. You see where there's not just an either or. It's not like there's just people that are tall and short in humans. We have a whole range of height that tends to usually form like a bell curve, where you've got a lot of people kind of in the middle, and then it tapers off as you get to the extremes. So if you're looking between, say, 5 foot and maybe, 6'2" or something, you're going to have a lot of people. And then there are some that go all the way up to like, 8 foot or whatever else, and there's some that go down to like 3 foot or whatever it is we're going to use in our range. But you'll tend to see this bell curve. It's not like with the pea plants where they were tall or they were short, where it was very clear which of these two things. And so you can see with these mussel shells, all the variation that there is. This is polygenic, and you can just see all these differences. There's not just like 2 types of their shells. There are tons of different types of their cells because they all are coming down on the spectrum. And so their shells have this whole gradient, if you will, of possible phenotypes. And if you see a gradient of phenotypes, it's polygenic. Now, for our fun stuff, except not fun, kind of sad, we have human disorders. So we're going to talk about recessive disorders first, which are the most common, because things that are really dangerous, if they're dominant, you tend to get them, express them, and then have problems. If you have problems and you can't reproduce, you can't pass it on. Because recessives can hide in carriers or heterozygotes, because of this ability for them not to be seen but to still be there, recessive disorders tend to perpetuate themselves better. And so cystic fibrosis is one you commonly see in Europeans. This is the result of a faulty chloride receptor that allows chlorine, I believe the chlorine channels is what it goes through to transfer chlorine in and out of the cell. So that screws up and it leads to an excess of mucus, which really just messes with your digestive system and your respiratory system. And so people who have this try to treat for the extra mucus. They can now live pretty long lives. I think a lot of them make it in like their 50s. It used to be like the 20s, but they've prolonged it now. But this excess mucus can then lead to digestive problems and respiratory problems, and usually one of those can lead to death. So treatments can delay this though, but it just comes down to this screw up in this chloride receptors, chloride channel within cells. Tay-Sachs is a lipid metabolic disorder where you don't break down a specific lipid in the brain and it builds up and becomes toxic. So people with Tay-Sachs, and this is specific to, and I'm probably going to butcher the name, but Anasazi Jews, yeah, I know I said it wrong, but a specific group of Jewish population. And so individuals that have this, so they're homozygous for this, will really like prematurely age. They'll be balding, they'll look older even at a young age, and they end up dying from it after they go through a pretty rough process where they deteriorate as the toxins build up. And so I believe they die usually around, say, like age 4. So this is a very rough disease because it goes through and it kills a child, and it's not at all even, not that there's a pleasing way to die, but it's a very prolonged process where they gradually get worse and worse. So it's not something that's very fun. And so if you come from a background that happens to have a propensity of Tay-Sachs, you can get tested. And so there are in some cases where people use this stuff to make decisions about whether or not to have kids, because if they're both carriers, there's a 25% chance they could have a child that has that. So it's something that certainly for at least one group of people they have to really think about. And then sickle cell I threw on here with an asterisk because it's kind of a oddball. I put it here just because its inheritance pattern is recessive, in the sense that you only get sickle cell disease or sickle cell anemia if you have, kind of, we'll do 2 lowercase here, if you're homozygous recessive. So because of that, even though it's actually codominant, because you only get the effect, you only get the negatives if you are homozygous recessive only, its inheritance pattern will be the same as a recessive disorder. So while it is caused by a codominant gene, we'll treat it like homozygous recessive because of that. And so we've already talked about how this one will impair a bunch of stuff within your body because of those sickle-shaped cells, and this one will affect primarily people of certain parts of African heritage, certain parts of Latin America, Basically, anywhere where malaria was pretty common, you tend to see this more because it is beneficial if you happen to be that heterozygote. Now, more interestingly, we've got some dominant disorders, which you've already said are rare and should be rare. But there are some cases where a dominant disorder can slip through. Huntington's works because if you get it, it doesn't actually show itself till you're typically around, say, 35 to 55. It varies fairly wildly in some cases, depending. But you don't start to get where your neurological functions start to kind of dissipate. You start to have control issues with your body, with your mind. And so you kind of deteriorate after that point till death. So Huntington's is a rough one just because you tend to have reproduced before symptoms occur. And so if this one occurred earlier on in life, you wouldn't reproduce and it'd just disappear. It'd kill itself off, if you will. But because it occurs late in life, that's one of the options for dominant disorders for them to work, is just to occur after reproduction. So I don't know I have it, so I can't choose not to have kids, or other people around me can't use that to decide not to mate with me. And then achondroplasia is a type of dwarfism where you tend to have pretty much regular sized torso. Your head will at least appear, it's kind of a little bit larger relatively speaking, but you tend to have smaller limbs because the cartilaginous plates, my God, that's terrible. It's kind of a turtle, but it's meant to be just showing short limbs. So if you have achondroplasia, it's where the cartilaginous, that's the chondral part, the cartilaginous plates in your bones and your arms and legs don't develop properly. They don't occur, they seal up early, and so your arms and legs don't grow to full height. Now this one, while there are some health conditions that do come along with it that make you more predisposed to issues, it's not like it necessarily has to kill you in some way. And so if individuals reproduce, they are capable then of passing this on, even though it's dominant. So this is one of those cases where it's a dominant disorder, but it's not so bad that it's making it where you can't reproduce, and so it's able to be passed on. Now, if you know that there's something in your family, there's several ways of kind of going through and trying to decide, should I do this? Should I have kids? And so one of them that's most used today is going to be just doing testing. Chorionic villus samplings, when you take a piece of the placenta, and they normally will do this where they go in vaginally through the cervix, and then they kind of suction off a little piece that sticks off the placenta that's made of the cells of the baby, the fetus. And they typically do this early. So this one's done like 8 to, at the latest, probably 15 weeks. It's usually more like 8 to 12. And so they're going to go through and take this sample, and then they can karyotype it, and they can genetically test it to see if something's wrong. And then if there is something wrong, they can try to either treat the child, or if it's something where it's going to be very dangerous, you'd have where you might get the option if you want to continue with the pregnancy, but they can try to figure out what's going on. And then amniocentesis is one that's typically used later. So this one's usually more like 18 to 22 weeks that they do this. And this is going to be a large needle. They use ultrasound to make sure where they're poking, and they're going to go through the abdomen into the amniotic fluid that surrounds the baby. That's like the water sac. That's what flows out when your water breaks. And so they're going to take some of this because just like we normally have skin cells slough off constantly, the baby does too. So by taking some of that fluid, they can get some access to the cells of the child, and they can once again run genetic tests, and they can run karyotypes to try to see if there's some type of issue. So you would have an option to figure out what you're going to do. You would have options if need be where it could be a defect where they can do surgery, or they just have to be very careful, or if you're going to continue, if it's going to be something that's lethal. So those are two of the common tests. Now the old school low-tech way that we still use for a lot of stuff is pedigrees. And this is just where you track your family by looking at what individuals within the person's past. And it could be a dog. You have pedigrees for dogs to make sure that they're healthy. This could be a person. It could be livestock. But you're tracking their relatives and whether or not they have a condition to try to figure out what the genotype is of a specific individual. So this way you could find out if you are homozygous recessive, hopefully heterozygous, or homozygous dominant. You want to figure out which one you are so you can try to figure out if it's okay for you to have kids, or if you're talking about dogs or livestock, if they're going to reproduce and give you the type of animals that you want consistently. So they can go through and say, all right, in this case, red's affected. And so depending on exactly what type of trait this is, looking at this one appears to be dominant because you'll notice it doesn't skip any generations. One parent always has it if they pass it on. So this appears to be a dominant condition. And so you can go through that and say, well, this person must be homozygous recessive. The fact that they had some offspring that don't have it means that this individual must be a heterozygote. And so you can just work your way down then, where if this person's a heterozygote and this person's homozygous recessive, all the offspring have to have at least one recessive. So this person must be a heterozygote, this person must be homozygous recessive, this person must be a heterozygote, and so you can just kind of work your way across and just keep filling these in. And as we keep going down, you can get to the point where you can start to say, all right, well then this is what this offspring must be. Because we already said this person must be homozygous recessive because they don't have it, and it's a dominant trait. So that means their offspring, once again, have to either be heterozygotes or homozygous recessive. So if we were trying to look at one of these individuals, we could fairly easily figure out exactly what their specific genotype is based on their phenotype. Now, if you've got something like Huntington's, this gets to be a bit trickier because you could figure out that you have a 50-50 shot if you know that one parent's heterozygous and one is homozygous recessive. But until the actual symptoms express, I don't know which one you are. So I could say it's a 50-50 shot. I mean, I could go through and show you the Punnett square and let you see, but we wouldn't know for sure unless you did more specific testing that could actually test for your alleles to see if you have the Huntington allele prior to expression. And for several disorders, we do have tests like that that can be done if they think that the person might have this disorder if it runs in the family. But this one doesn't require any fancy testing or anything else. And this can be done fairly easily for a lot of recessive traits that show up early. It's not a too difficult of a thing. And it can help you figure out, even if you have the disorder, it can at least help you figure out, are you likely homozygous dominant, heterozygous, and then you can figure out the odds of what's going to happen if you have a child with your spouse. And you can decide if it's 25% chance that they have an issue, a 50% chance, 100% chance. You can decide whether it's worth it or not to go through and reproduce with that person. Or maybe it's a good chance to adopt or try another method. You could even do something like surrogacy, where it's somebody else's egg or somebody else's sperm to try and prevent having a child being born that has the condition. So that gets into some of the ethics that each parent will have to choose if they get put in this particular position. Hope you guys have enjoyed this one. I'll see you guys tomorrow.